So we're moving on now to a discussion about how plants transport nutrients around within their bodies. And although there's both xylem and phloem that are performing these functions, this presentation is going to focus mostly on xylem. This basic figure is showing us water being taken up by the roots of plants, some oxygen being absorbed by the roots, carbon dioxide entering the leaves. Before we can really understand the mechanics of how that happens and how that water, for example, would get up to the leaves, we need to know a little bit more about the anatomy of the vascular tissue and the leaves. Xylem is the vascular tissue that's responsible for conducting water and the minerals and various solutes dissolved in water up the plant from the roots where the water is absorbed to the shoot system. Xylem as a tissue contains two kinds of cells. The first is called the tracheids. Tracheids are the more ancestral type of xylem. When we go back in the fossil record and look at the very first vascular plants, they had tracheids. Tracheids are tapering cells, they're fattest in the middle, and they come to a point at the ends. Vessel elements are the more derived type of xylem. They have generally a larger diameter and are more cylindrical in shape. So here is an electron micrograph of wood. This would be secondary xylem. And we see on the right some tracheids and on the left some vessel elements. So while these cells are growing, they form connections to their neighbors. Their shape is formed, their cell wall is laid down, but then the cells die and all the organelles and cytoplasms are resorbed and they become hollow tubes, conduits for the conduction of water. That's a weird thing about xylem is it's really only functional when it's dead. So the cell walls are what are useful here. The cell walls create these tiny vessels through which water and minerals are conducted. Phloem, by contrast, is only functional when it's alive. And phloem is transporting sugars and sugary solutions in all directions. They're not limited to just conducting up the plant. The cells through which solutions are conducted are called sieve tube cells. And like vessel elements, they're kind of stacked on top of each other. And the solution moves from one sieve tube cell into the next. As you know, the nucleus is the biggest, most conspicuous structure in most cells. So if a sieve tube cell had a nucleus, that would be an obstruction to the flow of solutions. So phloem cells are weird in that the sieve tube cells don't have their own nucleus. The nucleus basically lives in exile. It's contained within a little companion cell. So each sieve tube cell would have a companion cell where the nucleus is located. The end walls of the sieve tube cell are perforated, so solutions can flow easily from one cell into the next. Here's an electron micrograph. In the center we see two sieve tube cells. Their plates, their end walls bump against each other. We've got these perforations, these pores or plasmodesmata through which the solutions can flow. As I mentioned previously, xylem and phloem are bundled up together into packages we call vascular bundles. So xylem and phloem is always a package deal. And these bundles of vascular tissue have different patterns within the root and shoot. So if you follow this figure down, we see how in the shoot system of a dicot, the vascular bundles would be arranged in a circle. But by the time it reaches the root, all of those bundles converge to make one cylinder in the center. There's also differences between monocots and dicots. So there we see a side-by-side of a monocot and dicot stem, and the differences in the patterns of distribution of the vascular bundle. Understanding nutrient uptake and transport in plants really requires that you learned something in Biology 101 about membranes and membrane transport. So you may want to go back and refresh yourself in Chapter 5 and look at the images about solute transport. Make sure you understand, for example, active transport, how membrane proteins called pumps use ATP to transport solutes across a membrane, how channels allow for facilitated diffusion of solutes. You also need to understand osmosis, the diffusion of water across a membrane, how water molecules can diffuse across membranes based on solute concentrations, how water will diffuse from an area where solute concentration is lower, therefore water concentration is higher, to areas where solute concentration is higher, therefore water concentration is lower. So the diffusion of water is really dictated by solute concentration gradients. So the remainder of this presentation is going to focus on how xylem works. How can a plant transport water up from roots to leaves if plants don't have a pump? Right? We have a heart. Our heart beats to circulate our blood. Plants, like the tree in this image, can transport water up tens of feet high without any kind of pump. Plants are relying on more passive forces, utilizing a combination of forces that we collectively refer to as the transpiration, cohesion, tension mechanism. So transport through xylem begins as the plant takes up water from the soil by the roots. Roots have microscopic extensions, chains called root hairs that are only one cell layer thick. And that provides a lot of surface area contact between the soil and the plant. So as that plant is absorbing water, it creates a little bit of hydrostatic pressure in the root tissue, right? So there's a buildup of water pressure in the root that provides just a little bit of push. Now that sounds like that maybe that's an important force, but really it's a pretty minimal part of it. That hydrostatic pressure in the roots is referred to as root pressure. So our illustration here is showing us how water is following a couple of different routes from the root hairs to reach the xylem in the center of the root. So the water molecules can pass through the cell, being part of the cytoplasm, going through plasmodesmata from cell to cell. That's called the symplastic route. Or water molecules can be making their way through the matrix of the cell wall and squeezing their way in between cells. That's called the apoplastic route. So here we see water molecules moving through the root tissue on their way to the xylem. Their water molecules are moving through the matrix of the cell wall. They are passing through plasmodesmata from cell to cell, or they are crossing membranes, working their way through the matrix of the cell wall, going back through a membrane again, a transmembrane route. Now, once that water is absorbed and the xylem fills up, when you have a volume of water in xylem, it creates cohesion and adhesion. Those are two of the important emergent properties of water. Cohesion is the tendency of water molecules to stick together, and adhesion is the tendency of water molecules to cling to surfaces. So between these two properties, a unit of water inside of xylem acts together. All the water acts as a unit. So xylem in a stem is pipe full of water. There's a standing column of water, and any force you place on water, the whole column will react. If there's a push at the bottom, the column will slide up. If there's a pull at the top, the column will slide up. There's also a little bit of capillary action going on as well. So this is the natural tendency of water molecules to creep up narrow cylinders. You may have noticed this if you put a slender straw in a glass of water, how it creeps up above the level of the water. This is a really important figure that shows us that along the length of the xylem in a plant, there is a gradient of water potential. Water potential is a force expressed in megapascals. That is an SI unit of force. And we see that in the soil, the water potential in the soil is minus 0.3 megapascals. And the water potential in the air is minus 100 megapascals. So all along the length of the plant stem, there is a gradient that favors water moving up from the higher water potential in the soil to the lower water potential in the air. On the right side of this figure, we see how root hairs are in contact with soil water. And those water molecules are entering the root, passing through the ground tissue, getting into the xylem. Within the xylem, we have a standing column of water molecules. Each water molecule is hydrogen bonding to its neighbor, cohesion. It's sticking to the walls of the xylem, adhesion. That prevents that water column from backsliding. At the leaf end, this is perhaps the most important part of all this. At the leaf end, we have a process called transpiration. Transpiration is the evaporative loss of water through the leaf. And on the lower epidermis of this leaf are stoma or stomata. And when they're open to allow CO2 to enter, water molecules are diffusing out. And that loss of water at the leaf end creates a drop in water potential that helps pull more water up from below. So transpiration really is a key force in xylem function. The evaporative loss of water at the leaf end creates a drop in water potential that pulls at the top of the water column and helps water slide up the xylem. So take another careful look at this figure 29.18. It's on page 611 in your textbook. I think it does a pretty good job of showing us the forces involved. You should be able to describe this figure, discuss these forces, how we have this water potential gradient all along the network of xylem that make up the stem of a plant. We have water molecules clinging together to create a column of water that stick to the xylem. How transpiration at the leaf end results in a drop in water potential that helps pull water up. The key in xylem is up. Water flows up xylem. It's one way only. Here's our last image in this presentation about xylem. We have a cross-section through a leaf, an upper and a lower epidermis. We've got stoma, a single stoma on the lower epidermis. Each water molecule is a blue dot. So we've got water in the xylem in a leaf vein. And that water enters the airspace of the spongy mesophyll. And as long as that stoma is open, those water molecules are going to escape. The humidity in the spongy mesophyll of a leaf is 100%. There's a thin coating of water on all the surfaces. And as long as that stoma is open, water will evaporate. And that evaporation of water creates, again, a drop in water potential that helps to pull water up from lower in the xylem column.